Hi class, this is your chapter 14 lecture. The topic for today is cardiovascular physiology. We will go through 2 chapters to complete our section on cardiovascular physio. So this is the first. We're going through this entire chapter, but also check out some of these, this website here, as well as the ones that I put up in your chapter 14 folder. There's some pretty good online resources to help visualize these processes. All right, well, our cardiovascular system, just to begin, you probably know this already. It consists of our heart that functions as a pump, plus the fluid, the blood that circulates within the blood vessels. So the cardiovascular system does include the vessels as well, including the very plentiful capillaries. So we've got our heart, our blood, and then the vessels that distribute all the nutrients that go through our blood. So let's break down this distribution a little bit. So we have different levels of interaction with the environment, if you will. We'll see this also when we go into our respiratory section. But when we're looking at the transport of material, right, it does include the nutrients, water, and gases. But we also need to recognize that the bloodstream, the cardiovascular system, is also used to distribute all of the various hormones that our body uses. They all travel through, the water-soluble ones do travel through the bloodstream, but they target the cells that have their receptors. Our immune system cells and products of the immune system, such as the antibody, that too does travel through the cardiovascular system. We need to recognize as well that the cardiovascular system is important in removing metabolic materials, waste products, and other materials that cells no longer need. So it's not a matter of just delivering the components that our body needs. It also includes the removal of carbon dioxide, metabolic waste products, and even heat. So our blood vessels, we have arteries and veins. We'll spend more time in chapter 15 talking about that. But we've got different types of arteries, different types of veins. The capillaries are really where that exchange happens. And the design of our cardiovascular system is such that every single cell is able to receive the nutrients that do distribute through the cardiovascular system. Now, a portal system, this is something that we see in a few places in the body, but what a portal system is it is 2 different capillary beds that are joined. So it's a series of capillary beds. We have this in our kidneys, we have it in our intestine, in parts of our brain. So I'll highlight the important portal systems as we go through them. Of course, we have our heart, and the heart is divided by the septum into the left and the right heart, which have drastically different functions. The atrium receives the blood that comes back into the heart, where the ventricle pumps it out. Now, our blood is divided into the plasma and then the cells that are in it. So plasma is the liquid component of the blood. And then also another way a useful way to think of the cardiovascular system is that we have a circuit that goes through the lungs. That's the pulmonary circuit, versus the systemic circuit that pumps out into our out of the heart into the arteries. So we have pulmonary arteries and pulmonary veins. And then we'll look at some of the blood vessels, again, more in chapter 15. We'll spend the blood time learning about our different blood vessels then. So if we take all of the cardiovascular system, out of a body and then represent the connections, this is what it would look like, a simplified version of it. So here's the heart divided into the left heart versus the right heart. Yes, the left ventricle is actually what looks like on the right side here. But keep in mind that when we look at all of these images, the pictures are in what are called anatomical position. Anatomical position is when you're basically laying flat down on your back. So if I turn around and orient my back to this picture as it would be in anatomical position, right, my left heart is on the left side of me now. So keep in mind that when we look face down, we're not looking at the right perspective. You have to view it from, looking out of the body. So our systemic circuit begins with the pumping of that left ventricle that then sends the blood up through the aorta, where it then either ascends or descends into the aorta, going to roughly above the heart and below the heart. The arteries get smaller and smaller and eventually become the capillary beds, which are the sites of exchange. So this is where oxygen and carbon dioxide are exchanged at the level of the cell. It happens in the very thin-walled capillaries. And then that deoxygenated blood is returned through the veins back into the right side of the heart. When we pump out of the right ventricle, it goes up into this pulmonary circuit. So the right side of the heart pumps through the pulmonary circuit, where the left side of the heart supplies the body. That is the systemic circuit. So we've got very extensive capillary systems in our head and in our brain, as well as throughout the rest of our body. The lungs, as you may guess, are very well vascularized since the exchange of the respiratory gases happens there, right? We need to have the right blood vessels to facilitate this exchange. Now, if we look down at the bottom, we can see two examples of portal systems. So we've got the hepatic portal system, which connects the digestive tract with the liver. And then we also have a portal system in the kidneys. Both of these sites are major sites of exchange, which is why we see the portal systems placed here. Okay, so the vena cava are what bring the blood back into the right heart. We have a superior vena cava that drains from the heart above. And then the inferior vena cava is what returns blood from below the level of the heart back into the atrium. Okay, so now let's look at it a little bit differently. Blood, just like other types of material, it follows a pressure gradient through the cardiovascular system. And that pressure is generated from the pump function of the heart. So what we're looking at are the different types of vessels. So the aorta is closest to the heart. It branches into the arteries, which then branch to what are called arterioles, which then feed into the capillaries. And now we're going to turn from the oxygen-rich part of the way we can look at it to the more oxygen-deprived side of the vessels. So after the blood goes through those capillaries, oxygen moves out of the bloodstream to where it can be utilized. Right, we have a lot less oxygen there, and continues until we get back where it drains back into the right side of the heart, so that it can then go to the lungs and get more oxygen. So what we're looking at is a graph of the mean systemic blood pressure versus the different types of vessels that we have. And these are ordered in progression of how blood would flow. If you look at one drop of blood flowing from the heart all the way back to the heart, it would go through this pathway. So the arteries have a lot of pressure associated with them. And this is because they're right next to the heart. That's part of it. But it also has to do with the materials that these vessels are made with. But as we move away from the heart, the pressure declines down to almost nothing where it is returned to the heart. So we do have a pressure gradient. And as you know, gradients are important in helping the diffusion of materials, and our respiratory gases do move by diffusion. So the design of our cardiovascular system does match its function. Okay, so let's look at the physics of fluid flow first, and then we'll apply this to the cardiovascular system. On the left side of the figure, we're looking at this type of tubing that has, first of all, it's stopped at the end here. There's a stopper in here. And you can see that the level of water in each of these 4 cylinders is the same height since it's balanced out. These little arrows that that are pointing outwards, what these represent is the hydrostatic pressure, and that's water pressure. And it's proportional to the height of the water column. Now, once the cork is removed and the fluid drains out, then we see a gradient of that pressure having to do with distance. So there is a relationship between distance and pressure. So we have a drop off of it, and the columns that are closest to the exit or the opening have the lower water volume in it. So now notice that our arrows are pointing a different direction. We've got the flow of water out. So we see that as the fluid flows through the closed system, which our respiratory system is, or sorry, our circulatory system is closed, there is a proportional decrease in pressure. And this is because of distance and friction as the blood moves away from the heart. So flow is equal to the change in pressure divided by the resistive forces. This delta sign means change. It's the Greek symbol for change in. So I'd like for you to think about some of the factors that might affect resistance. Things like plaque being present inside a blood vessel, or the narrowing, simply the narrowing of a blood vessel. That has a relationship to pressure, flow, and resistance. We've got a number of different factors. Really, anything that changes pressure or changes flow will impact, is related to the resistance, right? We've got a relationship between these 3 factors. So the flow is equal to the change in pressure divided by the resistance. I know there's a lot on the slide, and usually I have the animations in here where I don't have everything coming up at the same time, but I didn't do that in today's lecture. So we'll just kind of go through each of these points one at a time. So blood flow, or any type of flow for that matter, airflow, is inversely proportional to resistance. And that, simply has to do with the setup of this equation. So flow is inversely associated with resistance. So as the flow decreases, resistance increases. That is what an inverse relationship is. So what Pascal's law on flow, fluid dynamics, is that the resistance is tied to the length of the blood vessel. So we see resistance getting higher as the length increases. That means we need to have more pressure to push it over greater lengths. Things like thick blood are a factor. So viscosity is a measure of thickness of a fluid. Water has no viscosity, where blood is viscous. Something like honey is even more viscous. So it's just the thickness of the blood. But this is tied to resistance as well. So the more viscous the blood is, the higher the resistance there is. So we can rearrange these formulas mathematically. I am more concerned that you understand the relationship between flow, pressure, and resistance. We will look at this part in just a moment, the relationship between the radius or diameter of the blood flow. And some of our blood vessels have muscle around them. So that means they are able to constrict or dilate under the right stimuli. Again, the flow is equal to the change in pressure divided by those resistive forces, whatever they may be. As flow increases, our pressure gradient is directly proportional. Our resistance is inversely proportional, right? Directly proportional is pressure to flow because they're both on the top. The inverse relationship is between resistance and flow. Okay, so now let's look at a few things. Let's look at the change in pressure first, and then we're going to look at changing the diameter. So we've got a pipe, call it a blood vessel, call it a pipe, whatever it is. In this system, we have flow through here. But now let's look down at the bottom. So put the numbers in so it makes it a little bit easier for us to understand. We have 100 millimeters of mercury at this side of this tube. There is 100 millimeters of mercury at the opposite side of the tube. There is no change in pressure. 100 to 100, there's no change. So this means there's no flow because there's no pressure gradient. We have to have a pressure gradient in order for there to be blood flow. Let's look at the right side. So now we have a gradient. It begins with 100 millimeters of mercury and ends with 75. So we have a 25 millimeter difference in pressure, in the pressure. So there is going to be blood flow. Let's look at the other example on the right side. So now we have quite different pressures, 40 millimeters of mercury versus 15. But look, the difference between the two is the same. It is 25 millimeters of mercury. So the flow is actually equal, even though it's different, because flow is proportional to the change in pressure, which is the same for both of these systems, 25 millimeters of mercury. So we've got equal pressure. So the flow is the same, providing there's no difference in resistance. Okay, so I've put in a few kind of questions in here, trying to get you to think about what to anticipate on your quiz and exam. Excuse me. So in order to maintain a constant flow, a constant blood flow, there has to be a difference between that pressure gradient and resistance. We have to overcome all those resistive forces, in other words, for them to be flow. So our blood moves out of the right ventricle. Where's it going? Out of the right ventricle. That means it's going to the lungs. Right side goes to the lungs, left side goes to the aorta. So in this example, you're told that the ventricle needs 50 millimeters of blood every minute to maintain its function. And the diameter of the right coronary artery, you're told, is 2 millimeters. Through the process of atherosclerosis, it causes narrowing of the diameter to 1 millimeter. So it's half the size. So what would we expect to happen to the resistance? Is it proportional? Because is it proportional to the change in radius? So, and it is. In order to maintain a constant flow, we have to have that ratio between the pressure gradient and resistant constant. So in this scenario, the resistance will double. If we take these numbers and plot them in, the resistance will double because we have decreased the diameter by half, or the radius by half. So another question, I've tried to separate the questions from the answers. So this is another question. The answer is on the next slide. So what happens if we double the resistance? What has to happen to the pressure gradient to keep the flow constant? So let's plug that in. So here's our formula. Flow is equal to the change in pressure divided by resistance. So we're told that, so that's normal is 50 millimeters per minute. But what happens with atherosclerosis? So what we've done is we've taken the flow, this is it, 50 mils of blood per minute is equal to the change in pressure divided by resistance. And now we're just solving, we're algebraically rearranging that equation so that we can solve for the change in pressure when we double the resistance. So let's take a look at how, we'll look at that in the next slide. So what we're getting at is a concept called cardiac output, which is simply equal to the heart rate times what is called the stroke volume. We'll learn about that. You'll understand that by the time we get through these chapters. But I want to introduce the formula a little earlier, just so we can start to learn how to use these words and know what they mean. So getting back to our example, here we go. So here's that scenario. So we're looking at 2 points here. Point A, the diameter is 1 centimeter squared. At Y, it is 12 centimeters squared. And so we've got a difference of 12 between point X and Y. So our flow rate would be 12 centimeters cubed per minute. Now let's plot it in. So we're looking at velocity, which is a rate, flow rate divided by area. Let's plug in our numbers. So we've got at point X, we have 12 centimeters cubed per minute divided by its radius, which is 1. So our velocity is 12 centimeters per minute. But at point Y, where it's much wider, look how much slower the velocity is. It's 1 centimeter per minute because it's so wide now. It's like a small lake feeding into a bigger body of water. There's so much space out there that it effectively dilutes the velocity or the flow. So our body has ways to deal with this in our normal physiology, but it also has ways to get us back to homeostasis in the event that we're off track. So the take-home message here is the narrower the vessel, the faster the flow rate. Another example you can think about just from your everyday life is if you have a hose, a garden hose, you turn it on, so no faucet or sprayer at the end, just a hose. You turn on that hose and the water comes out, right? It comes out at a whatever, related to how much water you put in there, how much pressure is going through it. But what happens if you squeeze the hose or step on it? We've all done this when we need to spray something off, right? We squeeze the hose or put our finger at the end and it increases. The water comes out, it feels like it's coming out faster or with more force. Well, it is. It is because we have increased the resistance by pinching down on that hose. It's moving through a narrower area, which means it's going through faster once we step on the hose versus release it. So again, just think about if you're doing something outside, say you need to hose off the fence or something, you can just, let the water trickle out of the hose. That's going to be a longer process than if you put your finger at the end and get that nice fast spray that comes out. It's more effective because the narrower the vessel, the faster the rate of flow. Okay, so why though does our blood flow so slowly through the capillaries? Because they're the narrowest vessels we have. But going back to that second figure we looked at, where we looked at the different blood vessels versus pressure, the pressure in the capillaries is pretty negligent. It's almost nothing. And that's necessary. Think about what would happen if the blood moved through so fast, there'd be no time for exchange. So it's necessary that the velocity, the rate of our blood flow, slow down at the capillaries so that the job of exchange may happen. So yes, there is a difference in the diameter of the capillaries versus the arteries, but there's also a difference in the materials that the vessel's made out of. Capillaries are just one epithelial layer. That's it. They're not surrounded by a bunch of muscle. What would that do? If there's a bunch of muscle beyond the capillary, it's not going to allow for exchange. So we have a very different job between our arteries versus the capillaries versus the veins. So we're going to learn about what those different jobs are with respect to the cardiovascular system. So these two processes, arterial sclerosis versus atherosclerosis, they're related. In fact, atherosclerosis is a type of arterial sclerosis, but they're different physiologies. Arteriosclerosis is hardening of the arteries, and this is usually because of a deposition of a mineral like calcium, calcification. But when the arteries harden, they have decreased compliance, this word compliance, as well as elasticity. We need to know what these words mean, okay? I'll explain it, but I'll also show it to you. So elasticity, you probably think of as stretching. Really, it's going back to normal after it's stretched. That's what elasticity is. So arterial sclerosis makes it so the heart has to work harder to maintain the flow because we can't adapt as easily to changes in the diameter to deal with pressure and resistance. Atherosclerosis is when materials like plaque build up in the wall. So underneath the endothelial layer, so inside the vessel. This too decreases the diameter of the vessel, but this is where we see a loss of elasticity and a risk of clotting can happen. Buildup of a plaque that eventually can lead to pieces of it breaking off, and in very bad cases that could travel to the lungs or to the brain and cause a blood clot there, which could be fatal. So we see these conditions more in elderly. We certainly see them more in people with inflammatory diseases like autoimmune diseases. Those are kind of classical examples of inflammatory disease. So there's a lot of good evidence now that shows using anti-inflammatory medications can have a benefit for the cardiovascular system. I need to qualify that though, because there's differences in different types of anti-inflammatories. And if you do any digging in this, you'll probably find quite easily that there was a huge malpractice, I guess is the way to describe it, situation having to do with anti-inflammatories and the heart. And unfortunately, some of the people that I worked with at the University of Arizona were involved in this and consequently lost their job because of it. But what they did is they started to administer some of these newer anti-inflammatory drugs to people with cardiovascular disease with the idea that it would help with the inflammatory component of the atherosclerosis. That makes sense. The problem is that this was done without having properly controlled trials, unfortunately. This was just in the early 2000s. It really wasn't that long ago, but it led to some really major changes in research design and also in publication rights. I worked with this person and I was told wrongly that because I used some of my research subjects were patients of this doctor's, they wanted me to put his name on my paper, even though this doctor didn't do anything to contribute to the research. And I didn't do that, but it happened with some other scientists. And unfortunately, the drug company basically said, hey, we want a medical doctor to be the first author on this paper, even though that medical doctor wasn't involved. And so it led to writing up of a paper of a very poorly controlled study where unfortunately 5 people died because of poor study design. So I'm bringing this up not really to share, a sad story, but to share with you that medical research is done well when it is done well, when the studies are set up properly. Just in the topic of atherosclerosis, there now have been a number of studies done that have shown that certain cholesterol lowering medications, the statins, do give a benefit. They do reduce cardiovascular disease risk in people that have inflammation. The type of the study I was referring to earlier was not on statins. It was on what are called COX-2 inhibitors. Celebrex, Vioxx are 2 of them. Vioxx was actually taken off of the market because of those 5 deaths. Okay, so now let's get back to the heart. So the heart has 4 chambers, and it is made up of a special muscle cell that's only found in the heart. It's made up of myocardium. And it is inside a special sac called the pericardium. So our heart sits inside a sac that helps it function properly. It also provides some protection, and that's the pericardium. Now, the atria sit on top of the ventricle. The ventricles are at the bottom and the atria sit up top, and they are thin-walled. Ventricles are very thick-walled. And specifically, that left ventricle that has to pump all out to the systemic circuit, it's even thicker because it needs the force that comes from that thick wall. So our blood vessels actually come out of the base of the heart. The aorta and pulmonary trunk take blood away from the heart. So the aorta goes to the systemic circulation. Pulmonary trunk goes to the lungs. Our vena cava, and this might seem weird, pulmonary veins return blood to the heart. So let's follow the path of deoxygenated blood. So it's moved out of the heart, gone through the arteries, gone through the capillary beds where that exchange has happened, into the veins, back up to the vena cava, where it's dumped into that right atrium, and then it moves down to the ventricle, and then pumps to the lungs so that it can get more oxygen and release any carbon dioxide. Our oxygenated blood goes from out of the lungs, so from the pulmonary vein. This is the part that's weird. Usually we think of veins as being oxygen little, right? We think of arteries as having more oxygen in them and veins as having less oxygen in them. Both of them have oxygen, but here it's different. The pulmonary vein is oxygenated. That's where the oxygen-rich supply is. It then goes to the atrium on the left side. It goes into the ventricle and then is pumped to the aorta. Now, our heart and the chambers have have connective tissue and some muscle around them, so that they can function in their right capacities. So we can look at some of this connective tissue really as just a marker, so that we can understand where the cardiac muscles are located. Okay, so here we go. Here's the heart. It's kind of weird, right? The apex, normally we think of a mountain. The apex is the point on the top, but here it's at the bottom. But it is still the point. Think of it like an upside down triangle. It is still a point. So even though it might seem weird that it's at the bottom, but it is. So here's the heart. We've got our two lungs. The diaphragm is necessary to generate the right pressure dynamics for both of our respiratory as well as our cardiovascular system. The thyroid is here. You can see these blue rings. These are the connective tissue rings that are around the trachea in this case. So these are good anatomical markers when studying this part of the body. Let's see, I'm gonna go ahead and move through. So in order for everything to work, there actually is a connection between all of these different structures. Let's see if I can think of a good example. Actually, let me just pause for one moment. I'm going to grab an example for you and I will be right back. Here's my example to show how the thoracic cavity works. So it's just really simple. I have just a Ziploc bag. It's got nothing in it, and I can open it, I can close it, but I can also pull the pieces together, the two sides of the bag, apart pretty easily. Now, in this one, I have some water. Get away from my computer in case I spill something. And where the water is, it's actually much harder to pull the bag apart. It doesn't come apart as well because the two sides are sticking together because of the pressure dynamics created in part from the presence of the water in the bag. So the thoracic cavity does just that. Let's get the slides back. By having the heart anchored so well in our chest by the diaphragm, by the muscles, by everything else in there that just wedges it together, that is part of what creates the setting for the pressure dynamics of our cardiovascular system. Okay, so now let's zoom in at the heart itself. So you can see the pericardium is being pulled away with some tweezers here in this cartoon example, exposing the heart itself. Look how big these vessels are that are coming out of the heart. You can see that the heart is attached. It is attached through connective tissue directly to the diaphragm. And that is what this simulation is supposed to represent, that it makes it hard to pull it apart when we have the pressure created in part from that hydrostatic pressure. So now let's zoom in a little bit more at the heart. Oh my gosh, Meatball's trying to get up here with me. Here he is. Look at him. Oh, he's going to jump right on the desk. Get ready. Come on over, Meatball. This is very weird. He doesn't usually do this. Okay, here's our, we're really just seeing the right side of it through this view, but the ventricle is at the bottom, the atria is at the top. So blood, deoxygenated blood comes in through the vena cava, superior if we're draining the head, inferior if we're below the heart. It dumps through the vena cava into the right atrium, where it then flows down into the right atria and then goes up and out through the pulmonary artery. Okay, look at the blood vessels that are present. And this is really just showing hardly any of them. The blood needs a good blood supply, so it is supplied with energy and nutrients to keep pumping constantly. So it's very well vascularized. So the ventricles do make up most of the heart, the bulk of the heart, and all of our arteries and veins attach at the base, which is at the top. Remember, upside down mountain, the apex is on the bottom, which means the base is actually up here. So the vessels attach at the base. Okay, so now we've cut part of the heart away so we can see in. And the flow through the heart is one directional, providing all the valves work properly. And when the valves start to be compromised, that's where we can see things like regurgitation, which is when blood goes backwards instead of moving forward. So let's run through the path. We're going to look at this one-way path. Let's begin where we just left off. So we've got our inferior and our superior and inferior vena cava that drain into the right atrium. The blood goes through a valve into the right ventricle, where it is then pumped through the coordinated contraction of that ventricle up through another valve, where it then goes to the pulmonary or goes out the pulmonary artery. Our other pathway is that the left pulmonary veins drain to the atrium, the left atrium, and then the blood goes through a valve where it passes into the left ventricle, where it is then pumped up and out through the aorta to the systemic circuit. So you can see the chambers. Here's the right ventricle, the left ventricle, the left atria, and the right atria. Now, our heart has two sets of different valves, and we have two in each set. So we've got a total of four valves, two different kinds. Look at these ones with this white kind of parachute looking material. So these are called the AV for atria to ventricle valve. On the right side, it also is called the tricuspid valve because it has 3 points of attachment. So all this white stuff here is actually part of the valve, but it is attached to muscles. See this little like red thing I'm trying to highlight here? That's called a papillary muscle. Here's the wording, papillary muscle. And the strings of the AV valves attach to these papillary muscles so that when the muscle contracts, it pulls the valve open, allowing the blood to move from the atria into the ventricle. The timing of this is important and is coordinated. I mean, look at just the different size between the atria and the ventricle. The ventricle can hold a lot more blood than does the atria, and that's true on both sides of the heart. So that means that in order for the ventricle to be completely filled so it can get its maximum function, these valves need to stay open so that blood can continue to come into the atria and then go into the ventricle, right? Because it's a much bigger volume here. So those are the AV valves. On the right side, it is called the tricuspid valve. On the left side, it is the bicuspid valve, and that has to do with the attachment points to those muscles. You can see the septum of the heart very well here, separating the left and right, or left and right chambers. The strings of the AV valves, these are called the chordae tendineae. And do think of it. I think a parachute is a good analogy here, where if we pull that muscle, right, it's going to pull those chordae tendineae, opening the valve. The other valves are at the exit points, okay? So we have, and these are called semiluminar valves. So we have the pulmonary semiluminar valve that is the connection between the right ventricle and the pulmonary artery. And then we have a similar valve at the aorta that separates the left ventricle from the aorta. So that valve has to be open to pump the blood out. Now looking at this little box, we're looking at a cartoon of the arrangement of the individual myocardial cells, those individual muscle cells that are only found here. This is a different type of muscle. So they're shaped like a Y, and they have these special strong connections called intercalated discs. That heart is constantly pumping, 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 pumping. So the evolution of this very strong junction fits with the form and function of the heart. It's a strong connection that's elastic, and what we don't see here is that there are many, many, many gap junctions present in the intercalated discs, because those ions that coordinate all of this need to flow readily over and through the heart very quickly so that it effectively functions as a single contraction. So myocardial muscle cells are branched. They have one nuclei per cell. That's different from skeletal muscle. And they are connected by these specialized junctions called intercalated discs that are found here in the heart. There are many mitochondria present in myocardial tissue so that it has energy to do its, to keep doing its job. Okay, so here's a little table that basically just organizes the flow of blood between the heart, the different chambers of the heart, and the 4 major vessels coming out or in, out of the heart. So we've got our right atrium receives blood from the vena cava, and then it goes through that AV valve down to the right ventricle. Our left atrium receives blood from the pulmonary veins, and then it goes to the left ventricle where it can be pumped out. The left atrium then drains the blood into the left ventricle where it then can be pumped out. So please refer to this in your own time. And I would encourage you to make a drawing of it. That will help to cement the connections for you. In fact, I have a figure in here towards the end of the slide set of the heart and the vessels so that you can practice labeling and drawing the pathway of blood. Okay, so let's summarize those valves again so that you can see the names. So we have 2 types. We've got the atrioventricular valves, the AV valves that separate atria from ventricle. And then we have the semiluminar valves, and we have 2 of each type. We've got the chordae tendineae, and these are those strings that attach to the papillary muscles. The right AV valve is the tricuspid valve. The left AV valve is the bicuspid, which is also known as the mitral valve. So many names. Just wait till we get to immunology. The semiluminar valves are between the ventricles and the arteries that they lead to. So we have the aortic semiluminar valve and then the pulmonary semiluminar valve. So we've talked so far about the systemic circuit of blood. We've talked about the pulmonary circuit of blood. What we haven't mentioned is the coronary circuit. So we do have 3 circuits of blood. The coronary circuit is incredibly important because it is what supplies blood to the heart so that the heart can do everything, right? It needs a constant supply of oxygen, of energy in order to function. So that's what the coronary circuit does, is it supplies the heart with the materials that it needs and takes away those metabolic waste products. So we have coronary arteries as well as coronary veins. And many people that have cardiovascular disease have it's associated with the coronary vessels. Okay, so looking at the flow, so we're looking at what happens in ventricular contraction, and we've got two different views of the same thing. So on the left side, we're looking at what's called the frontal section, where we're just looking flat in, but really, right, it would be anatomical position. And then we've got the cut, the transverse cut, which cuts it in half. So you can see the left atrium. Here is the mitral valve. The chordae tendineae are attached to the papillary muscle, which in this case is contracted. So it's right. You can see that it's closed. The left ventricle is contracted, and the AV valve is closed. The aortic semiluminar valve, though, is open. So what I'm trying to impress upon you is that these valves should not be open at the same time. The heart doesn't function properly if they are. In order for us to have the proper pressure dynamics, these valves need to operate properly. So the contraction of the ventricle is what pushes open the aortic semiluminar valve. There's enough pressure behind that contraction just to push it open so that the blood can move to the aorta. So during ventricular contraction, the AV valve is closed. That is very important. If the AV valve were open, blood would just continue to dump in from the atrium. We don't have the closed pressure dynamic anymore with that valve open. So what this does is it keeps the blood flowing in a one-way circuit, right? If this AV valve were open during contraction, blood would squeeze back to the atria. It would just move back the wrong way. It would regurgitate, and that is pathology. So looking at the transverse section, you can now appreciate that our AV valves are closed, both of them. So we've got our mitral, which is our left bicuspid, and our tricuspid, which is the right. They are both closed at the same time when our semiluminar valves are open. They should not be open at the same time. All right, so looking at the coronary circulation, and again, this is just a little drop in the bucket. The blood vessels go all over. We're just showing you the major ones, but they're extensive. There's a good arterial and venous side all through the heart. I'm not concerned that you learn the different blood vessels of the coronary circuit. That is anatomy. I want you to understand that we do have a coronary circuit and what it does. Okay. So that purpose is simply to supply the heart with what it needs and remove the waste products so that they don't build up. But just like we see in the rest of our body, there's a branching of sizes within the coronary circulation too, where the vessels start big and then they get smaller and smaller so that there can be exchange through those capillaries. Okay, so now let's get to the contraction part of it. And this is a big difference between cardiac muscle and skeletal muscle. Cardiac muscle doesn't get its signal from the nervous system. I mean, sure, it can be influenced, but the heart has its own special type of cell that provides that action potential to generate the contraction. So we have two types of cells in the heart. We have what are called the pacemakers. They are autorhythmic cells. These make up about 1% of the heart. 99% of it is contractile cells that the job is to pump, to contract. Contractile cells are organized as a sarcomere, just like we learned about with our skeletal muscle. The autorhythmic cells aren't, because their job is not to contract. They don't need a sarcomere because they don't contract. Instead, they provide the electrical signal, and we'll learn about how. So I wanted to give you a list here to summarize the main differences between cardiac muscle and skeletal muscle. So at this point now, you are responsible for knowing the features of the cardiac muscle. So cardiac muscle is smaller. It has one nucleus and it's shaped like a Y. The branches are connected through the intercalated discs that have lots of gap junctions so that our ions can flow readily all through the heart. The T tubules that are present in the contractile cells are bigger and they branch more. Again, T tubules are the connection to the outside. That is where all of those necessary ions and nutrients flow. So there need to be T tubules all through the heart so that every single heart cell, every muscle cell is bathed and accessible to the ions that are needed to initiate that contraction. The sarcoplasmic reticulum is smaller in cardiac muscle than it is in skeletal muscle. This does not imply that calcium is less important, though. Remember, calcium stores in the sarcoplasmic reticulum. Now, a third of the cell volume of the heart is just mitochondria. That's significant. We need lots of energy to keep that heart pumping. Okay. So the way let's look here. So look at the spiral arrangement of the muscle. It's all designed to squeeze from the apex at that bottom of the ventricle in a coordinated way so that it literally squeezes the blood up and out. So even the anatomy of the heart, oh, that bag's leaking. Even the anatomy of the heart facilitates its contraction to push the blood up and out of the heart. So now looking at an intercalated disc, you can see there's a lot to them. And these are very, very, very strong junctions. They have what is called a desmosome in it with the gap junction that allows for the rapid transfer and movement of the fluid. So we've got our one nucleus. You can see many mitochondria. Again, a third of the cell volume is mitochondria of the heart. We've got our intercalated discs and many gap junctions and desmosomes that go through the heart. So we have two types of heart cells. 99% of them are the contractile type of myocardium, where 1% is autorhythmic myocardium. They are both myocardium. That is the name for heart tissue. So please think about how these two types of cells, the contractile versus the autorhythmic cell, how are they similar and how are they different? And what differences exist between these two muscle cells versus the skeletal muscle? Now, our skeletal muscle sarcomeres do get fully activated when they're stimulated, meaning that all the myosin and actin are interacting. With our cardiac muscle, though, that's a little bit different. There's another role of calcium here. The calcium is important in helping to generate the contractions of the heart, just like it is for the skeletal muscle. But let me point out those big differences here. So one of the big differences between skeletal and cardiac muscle is the force of the contractility is proportional to the calcium, okay? We do have local and long distance controls. We have our long distance reflex controls through the action of a molecule, a neurotransmitter called norepinephrine. And then we have a neurohormone called epinephrine that are tied to that control loop. Locally within the heart, it's actually the stretch of that myocardium. And what that does is when it stretches, it increases the response or sensitivity of troponin to calcium, right? And we need that interaction to happen so that the actin and myosin are able to engage properly for the contraction. So far we've learned about the action potential of the neuron, of the skeletal muscle. Now we're going to learn about it for the contractile myocardium, as well as the autorhythmic myocardium. I suggest you make yourself a table so that you can see these comparisons. Okay, so what we're looking at now is the relationship between stretch and tension in skeletal and cardiac muscle. So on the x-axis, we're looking at the ratio between the initial length of the sarcomere and its percent of maximum, or percent maximum of sarcomere length. On the y-axis, we're looking at the percent maximum of tension. So the relationship between stretch and tension. So the solid line is skeletal muscle. The dotted line is cardiac muscle. And this kind of peachy shade area is the physiological range in which cardiac muscle is active. Look at the slopes of both of these curves. They're different. The skeletal muscle has a sharper curve, where the cardiac muscle doesn't. So most of the tension is associated with the physiological range in the cardiac muscle. That's not true in skeletal muscle. So we do have a relationship here between the stretch and the length of those sarcomeres. So with our skeletal muscle, its greatest change occurs between 60 to 80% of its maximum length. Not true for cardiac muscle. For cardiac muscle, its greatest rate of change is occurring between 80 to about 95% of its maximum length within that physiological range. Okay, so now we're going to go through the steps of the contraction of the heart muscle. And keep in mind that we have two different types of myocardium. So we're going to have to learn about both of them. But right now we're studying those contractile cells. So the action potential comes from the autorhythmic pacemaker cell right there in the heart. That actually saves time because it doesn't need to go through a reflex. It's right there within. So we call it a calcium-induced calcium release. This is different from our skeletal muscle. Calcium-induced calcium release. Okay. We're still talking about the same receptors, the RYR receptor, which is on the sarcoplasmic reticulum. So in our skeletal muscle, we looked at the movement of sodium as a early step. Here we're looking at a movement of calcium. So it's called the calcium spark. And that leads to a calcium signal. Again, calcium-induced calcium release. So a spark leads to the signal. We still have the same role that calcium binds to troponin, so that's the same. And the cross-bridge cycle is pretty similar as to what we saw from this point forward in skeletal muscle. Now we have some, we'll look at the differences. We've got another pump that we haven't learned about yet. So we have a calcium pump that's, we saw that with skeletal muscle to get our calcium back into the sarcoplasmic reticulum. But now we've got the sodium calcium exchanger. Let's take a look as we go through. Okay, so we're looking at the excitation coupling in the contractile cardiac muscle. So it begins with an action potential moving down that T-tubule. The action potential is what triggers the calcium channel to open. So the action potential, once we hit a certain electrical potential, the calcium channels open, allowing calcium to flow inside the muscle cell. So calcium flows in, and now at step 3, here is our calcium-induced calcium. The flow of calcium in is what activates the RYR receptor on the sarcoplasmic reticulum to open, which then releases a lot of calcium. So this is the calcium signal. Here's our calcium flow leading to our calcium spark, leading to our calcium signal. Sorry, I said that backwards a moment ago. So step 4, we're looking at our calcium spark, and it is summed. So that's why we see these multiple arrows converging into one. We'll get to that. So it's a summed signal. And now what happens once after the spark gets to the signal is the calcium then can go bind to troponin, which causes the tropomyosin to slip, exposing those actin and myosin regions to each other so that we can have contraction. So that gets us through the contraction. So calcium is necessary to initiate the contraction of cardiac muscle. The relaxation side of it is over here, starting at step 7. So what happens now is once the calcium unbinds from the troponin, so it's going away, it's moving back up through this calcium pump, returning it to the sarcoplasmic reticulum. Now we've got relaxation. So calcium pumps into the sarcoplasmic reticulum. We're going to move up to step 9 here. We also have calcium that goes through the sodium calcium exchanger. So calcium goes out to the extracellular environment and in comes 3 sodium. So 3 sodium come in, and now the sodium interact with the sodium potassium pump that we learned about earlier in the semester, where we take those 3 sodium and pump them out against their gradient. And in return, we bring in 2 potassiums against their gradient, which is why we need ATP. So our calcium is exchanged with sodium through the sodium calcium exchanger. But importantly, our sodium potassium pump maintains the sodium gradient so that we can get back to homeostasis. Okay. So we have a few phases in our process here. So this will make more sense when we look at the next graph, but I wanted to give you the wording so that you have very clear steps for each of these phases. So phase 4 is at resting. We depolarize and we've got sodium movement. I'll get to that. In phase 1, that's when we're repolarizing, our sodium channels close. We hit a plateau. This is new, we haven't seen this before. And then we enter the repolarization due to the changing in the different ion channels. So it creates a very different shape action potential curve. And again, this is still for the cardiac contractile cell. We're not talking about the pacers. But look at that plateau. So this is the shape of the contractile action potential. And this, so effectively, it's longer, right? We've got this plateau that spans a great period of the time. And as we'll look, we're going to look at the refractory periods. This long action potential is really important in preventing tetanus of the cardiac contractile cells. Think about what would happen if tetanus happened with the heart muscle. It wouldn't be able to pump. That would be potentially catastrophic. So the plateau, that long duration, is from step 2. So let's go through it. So this lines up with the slide previously. We're at resting membrane potential. Look where we are. It's a new number, -90 for cardiac contractile. It was -70 millivolts for our skeletal muscle and for our neuron, but now we're at -90 thereabouts. So we have the opening of the sodium channel, which leads to the rapid depolarization, bringing us up to 0.1. The sodium channels close. And now what's happening in plateau 2 is the calcium channels open, and some of our potassium channels, the ones that move it through fastly, they're closing. So that gives this plateau. Next, what happens at 0.3 is now the calcium channels are going to close and our slow releasing potassium channels open, and that gets us back to resting membrane potential. So the time of action potential is somewhere about 250 milliseconds for the cardiac contractile cells. All right, so now let's overlay the action potential with the actual muscle twitch. Okay, so the action potential of the contractile cell is in red, and then the muscle contraction is in blue. This yellow is the refractory period. So it spans almost the entire time, preventing tetanus, very importantly. So first we have to have the action potential fired, right? Just like we saw with skeletal muscle, we have to have the signal first, and then comes the contraction. So we've got membrane potential on the y-axis, also tension. It depends what line you're looking at. So for the action potential, it's membrane potential. For the muscle, it's tension. So look over here in part B, we're looking at just the contraction cycle, but look at it. The refractory period is pretty much that whole time. We have very little time in between. And this is to prevent tetanus. It means that we can't stimulate another round until we're done with this first one. So this is necessary to prevent tetanus. Let's compare that with skeletal muscle. So here's the action potential of the skeletal muscle, followed by the contraction of skeletal muscle. And our refractory period for skeletal muscle is much shorter. It spans that action potential time, but not the muscle contraction. So this is why we can get summation and tetanus in skeletal muscle, but not in cardiac muscle. It's not possible to sum leading to tetanus in cardiac muscle. So if we hit the skeletal muscle over and over again with a frequent stimulus, it can get up to tension, to tetanus, to fuse tetanus. But that doesn't happen because of the difference in the refractory periods. Okay, now we're going to switch gears and begin our discussion of the autorhythmic pacemaker cells. So far in your table, you should have the neuron, you should have skeletal muscle, You should have the cardiac contractile cells and now the cardiac autorhythmic cells. And just make yourself a little Excel spreadsheet where you can put in the membrane potential, the time of refractory period, different things like that, when different channels are involved. Okay, so let's look here. So this is a unique shape too to the action potential. So we have what's called this pacemaker potential, where we're basically getting ready to go. So it leads up to the threshold of about -40, and then we get a sharper increase leading to that action potential. And then we go back down to our membrane potential of -60 millivolts for the pacemakers. Now let's look what's happening with the ions. So here we have a new channel here we haven't talked about. We have what are called these funny channels, IFs for ion funny channel. And yes, that is what they are named literally. That is what is the pacemaker potential is all about, is tied into those funny channels. So we have a flow of sodium in, and then we get to our threshold of -40, the calcium channels open, allowing calcium to rush in, and then we have the changing of what's happening in our gates. So our calcium channels close and our potassium channels open, which allows for the movement of potassium. And then finally, we get back down to membrane potential. So the funny channels are what are responsible for this period in between potential to threshold. And they're called funny channels because they're not as specific as our other channels. More than one ion can move through at different times. But these are incredibly important in generating that pacemaker potential. Without the funny channels, we don't have this pacemaker potential. So here we are again on the other side of it, getting back to threshold to go into the next action potential for these autorhythmic cardiac cells. So our autorhythmic cells we call it a pacemaker potential rather than a membrane potential. That's not really important to me. I'm okay with you calling it a membrane potential, but I want you to understand what's happening here. These funny channels are really important in it, leading to the depolarization. Okay, now let's add in another layer of complexity. Let's add in some effects from the nervous system. So we've got our normal action potential in gray. So it's kind of hard to just separate them out. So we've got 1 peak, 2 peaks, 3 peaks in our normal stimulation. But when we hit it with sympathetic stimulation coming from the autonomic nervous system, sympathetic increases. So now we've got 1, 2, 3, 4, 5, almost 5 action potentials happening in the same time frame of 2.4 seconds. So we've increased the action potential here by sympathetic stimulation. The heart will beat faster due to the sympathetic stimulation because we're firing more frequent action potentials. But I do want to point out, look what's happening. With sympathetic stimulation, we're not quite getting back down to membrane potential, right? It's above -60. So it's not quite getting all the way back down. So that is why it's able to fire quicker, because we don't have as much distance to cover, right? Going from here to potential or threshold is smaller than going from our resting to threshold. So our ventricle action potential is really limited due to the getting back to those funny channels again. So our maximum heart rate is about 240 beats per minute, which is very dangerously high. But in order for that to happen, there would be absolutely no time in between the action potentials, no time to recover. So it's going to be very difficult to function properly at that high stimulation. Oppositely, the parasympathetic stimulation would decrease the rate. So we still have our 3 peaks in the normal simulation, but now only 2 in the same time frame under parasympathetic stimulation. So and the reason why is it? Because we have this hyperpolarization. That's why. So with our sympathetic stimulation, we had a shorter period to go to threshold. Under parasympathetic, it's longer. We're below that resting value of -60. And look how much time it takes to get to threshold. So that's why it's slower. So parasympathetic stimulation does hyperpolarize the membrane potential of the autorhythmic cell. This is autorhythmic. All right. Now this is, this could actually start the table I'd like you to build. But we've got skeletal muscle, we've got our contractile heart, we've got our autorhythmic heart. And then I'd encourage you to maybe over here have a neuron. So we've got our membrane potential, we've got the different events associated with our depolarization, our repolarization, the action potential. Look at the time frame. Skeletal muscle is 1 to 2 milliseconds for the action potential, but for the cardiac contractile, it's 200 milliseconds or higher. With the autorhythmic, that can vary some, but it's about 150 milliseconds. We've got a very brief refractory period in skeletal muscle, but it's very long, almost the entire length of the action potential in our cardiac contractile muscle. With our autorhythmic cardiac cells, the refractory period, it's not the same because well, that's where it's all starting from, right? It's not as relevant because these aren't the cells that are contracting. Okay, so now let's take a look at the flow of that electrical current. It begins at a place here called the SA node. So if we're looking at the action potential of the autorhythmic cells, here's that shape. The contractile cells have that characteristic plateau. Okay, so hopefully you can recognize them just based on shape. So the electrical current begins at the SA node and then very quickly spreads because of the anatomy of the heart. It allows for the rapid movement of all of those ions through the intercalated discs that have all those gap junctions in them. So our autorhythmic cells are spontaneously firing their potential, and then that leads to the potential of the contractile cells as the ions move through those gap junctions. All right, so this is a figure I put in here so that you can have something blank to label. So the red points refer to the electrical points in the conduction system that we'll study. But as you can see, you've got the different vessels. Maybe you want to play around with labeling all of this and even drawing the flow of blood. So these extra points are other areas of the heart I'd encourage you to learn so that you can understand the function, the physiology. So everything in red, 1 through 6, is the electrical conduction, and then the rest are different anatomical areas. Okay, so now let's go through the conduction. So now we're looking at all these different regions, and please do look at the videos that I've put up for this. Sometimes it's easier to see it in a video than to look at the stationary images. It begins with firing of the SA node, which then travels through what are called the internodal pathways to get to the AV node. The figure reverse the letters here. It should be the AV node. So then from the AV node, the electrical signal goes through the AV bundle where it then branches out. So we've got two now. And then it moves down into what are called, through the bundle branches, it moves to what are called the Purkinje fibers, which are present in lots of area. Okay, so that's the overview of it. Now let's tie that into the activity of the heart in the different parts. So our SA node fires, it depolarizes. Quickly, that electrical activity goes through the internodal pathways to the AV node. So right now our atria would be active because the signal has not yet gotten into the ventricles. So this purple shading is the depolarization. It's showing you what part of the heart is depolarizing when. So as our signal goes to the AV node, it is then going to branch out through the bundle. So now we're starting to see full engagement of both atria. Look, our valves are open. Our AV valves are open. After the bundle branches, it moves down to the apex of the heart, where that contraction begins at the point at the apex of the ventricle, and then it moves up and out. Why does it move up and out? Because that's where the Purkinje fibers are located. So different parts of this electrical conduction system are active at slightly different times, but it is all tied to the contraction of that of the contractile myocardium, which begins at the base of the ventricle and then moves up and out, allowing all of the blood in the ventricle to be squeezed up and pumped out through the valves leading to either the pulmonary circuit or systemic circuit. So our depolarization wave spreads from the apex of the ventricles up towards the base. Okay, so now let's talk about the action potentials at different points in this conduction system. So the way I like to think about it, and your book describes it this way, it's like a game of follow the leader. So imagine we're all kids again and we're outside, and the leader of the group is the SA node. So the leader of the group is setting the rate at about 70 to 80 action potentials per minute. And as long as the SA node is still the leader of the group, everything's firing about the same. But what happens though if that person gets tired and they drop out? Then the second fastest person takes over leadership of the group. And that would be the AV node, but it fires much less. It's firing about 45 to 50 action potentials per minute. Now, if the AV node drops out, then the AV bundle becomes the leader. And now we're dangerously low. This is not high enough to support life. If the AV bundle drops out, then we get to the Purkinje fibers at only 35 action potentials per minute. So as long as the SA node is functioning properly, it does lead the pack. It does pace everything else. It is the pacemaker as long as it's working properly. And with it firing about 70 to 80 action potentials per minute, this is how we get to our average resting heart rate. All right. So now let's move to the electrocardiogram, which you've had some introduction to in lab, but let's get to the nitty-gritty of it. So you see this called an ECG. It's also called an EKG. Same thing, really. So first of all, what is an electrocardiogram? It is not an action potential. Okay, it's not. What it does is it shows the electrical activity from all of the cells in the heart, right? It's not that we're sticking a probe in one part of the heart and measuring just that response. I mean, that can be done. It's invasive. but we're looking at the summed activity of all of it. So we have three main waves in an EKG or ECG, and then two segments. The waves are the P, the QRS, and the T. Those are the three main waves, and they're associated with different activities of the heart. So the P wave is atria depolarization, right? That happens first, then the blood goes down into the ventricle, and then the ventricles start to depolarize. So the QRS is the ventricle depolarization. It's also, there's some overlap with the atria repolarization here too. So again, we're not looking at separate events with the EKG. We're looking at the sum. Now, the T wave is repolarization of the ventricle. The segments are main segments. We have the PR segment, and this is a way to look at what would be called the AV node delay, where the TP segment allows us to look at the ventricular relaxation as well as the atrial relaxation. Why? Let's look. Okay, so our, of course, the action potential of the pacers comes first. And then we get the contractile steps that come next. So here we go. So this is the typical pattern of an EKG. So we have our P is this hump, Q, R, S, T. P, Q, R, S, T. An electrocardiogram is done by putting electrodes on different parts of the body, but at minimum, we need to have a triangle. And this is simply so we have a direction and current to flow. So notice we've got positive on the left arm, and then we've got a difference in the flow. So positive, negative, positive, negative. And then positive, negative. So the positioning of the leads allows, we have one positive and one negative electrode, so that there's a difference in the electrical gradient so that we can measure it. Okay, looking a little bit closer. Our P wave, Q, R, S, and then T. Our PR interval is at the beginning of the P to through this period, so through the R, and then we have our QT interval, and then we have it again, another cycle of it. Let's see. So the way that I like to measure heart rate is actually from R to R. You can do it P to P, you can do it T to T, as long as you're in the same exact point in each of those waves. So if you're doing the P wave, it needs to be at the exact same point in the P wave between one cardiac cycle and the next. So for me, I like to go from R to R because it is a single point. The textbook describes it as measuring it really P to P, but R to R works just as well. In fact, I think it's a little easier to measure. I don't recommend using the Q wave for trying to figure out the heart rate. The reason why is some people don't have a Q. So it would be pretty hard to base a standard on it if you don't have it, okay? So the EKG is printed on this graph paper, and there's a relationship between the distance in the waves and the heart rate. I'll show you how to calculate that. So our P wave is the atria depolarizing. That happens first, and then the signal spreads out of the SA node. It's already happened now because we have our depolarization of the atria. So we're at least at the AV node moving into the AV bundle. So when we're looking at the PR segment, which is right over here, here's the PR segment. This allows us to determine if there are any issues with the conduction through the AV node and AV bundle. Whereas if we look at the ST segment, we're looking at repolarization of the ventricle. So by knowing what's happening at the different points in the EKG, we can infer different information about different parts of the heart in both contraction as well as the excitability coming from those pacemaker cells. So our PR segment is conduction down to the AV bundle. Our QRS complex, that's this big point, is ventricular depolarization. And then the T, this hump here, is the repolarization of the ventricle. Okay, repolarization of the ventricle. And notice that the T wave is bigger than the P wave. And it should be, because the ventricle is a lot stronger than the atria. Okay, so we can use the EKG, ECG to figure out different things. So this is the flow of what I recommend doing if you're analyzing an EKG. So first of all, we would ask, well, what's the heart rate? And we can calculate that from, like I was just saying, R wave to R wave, or P to P. And of course, the BioPac machine can do this for us as long as we tell it what points we're going through. So when we have a faster than normal heart rate, that's tachycardia, whereas a slower than normal heart rate would be bradycardia. So what's the heart rate? And then we look at the rhythm. Is the heart rhythm regular or irregular? Meaning, do we have all the different waves happening? Are they in recognizable form, right? Is the ventricular response bigger than the atria? It should be, because the ventricles are stronger and they're bigger. Is there a single QRS for each P wave? And then is that PR segment constant in length? So these are the things that clinicians are looking for when they interpret an EKG. And if we see changes in these parameters, that could suggest a pathology in either the conduction or the contraction, or both. Okay, so now let's tie together the different events on the EKG with the electrical conduction. So we're looking at the same figure we looked at a little while ago when we were studying the movement of the SA to the AV, to the AV bundle, to the bundle branches, down to the Purkinje fibers. But now we're overlaying it with the EKG. So we're going to begin up top with the SA node firing. There's step one. So let me point out the peach coloring in the graph or line, that represents, the repolarizing of the heart, where the purple is depolarizing. So keep in mind in an EKG, let me actually get back to the picture. There's overlap between depolarization and repolarization. That means you can't use the direction of movement as an indicator of exactly what's happening. When we were looking at in our nervous system, right, a depolarization always went up, where a hyperpolarization always went to a lower membrane potential. We can't say that simply here, because there's overlap between the atria depolarizing and then repolarizing, and then the ventricle doing the same. Okay, so we begin with the SA node firing. Now we get our depolarization of the atria, leading to our increase in the P wave. Next, what happens is the atria are going to contract. So we're in this little peach segment now. So we've completed our P wave. We've gone through the PR segment. That means that the electrical conduction has made it through the AV bundle. So SA node to the AV, out to the bundle, and then we're going to branch next. So now we're doing that little peak down, little drop is the Q wave. And this is from the repolarization of the atria. As we continue, now our atrial repolarization is complete. And now we've gone to the top of the R wave through the completion of that atria repolarization. As we continue, now the electrical system or conduction is moving through the branches, and we've gotten down through the completion of the QRS complex as the ventricles are depolarizing. Next, as the electrical conduction moves through the Purkinje fibers that are concentrated at the base and then, or excuse me, at the apex and then move up, our ventricles are contracting and we're having repolarization of the ventricles. You can see that little peach color here leading to the T wave. And finally, we get to the end and we're completely done. So the action potentials go from the apex of the ventricle up to the base. And that is what allows the contraction to follow the same pattern from the apex so that it can squeeze all of the blood up and out of the atria so that it is all expelled. So the atria are fully emptied out of their blood. And that allows us to get to the best next cycle. Okay, so now looking at this a little bit differently, and this is a little tricky. Please don't worry about this part of it yet. We'll talk about this more really as we move into the respiratory system. But looking at the contraction, so that's what systole is, where relaxation is diastole. So blood pressure is systole, contraction over relaxation. So let's start at stage 1. So we're going through one complete cardiac cycle here. So at stage 1, we are relaxed, everything's relaxed. The ventricles are going to just fill, and that happens passively as the blood is put into the atria. It will flow down to the ventricle. This is still in the diastole, so that's why it's passive. It's a passive filling. But as we move through the cardiac cycle and enter atrial systole, now our atria are contracting. That generates force that blows open those AV valves with the help of the papillary muscles contracting. So they pull down, opening the AV valve. And then we enter this period of time called isovolemic ventricular contraction. Boy, that's a mouthful. Isovolemic, same volume because the ventricles are full. So it's the same volume. We haven't gone out of the ventricles. We haven't opened up the semiluminar valves yet to pump out to the rest of the body. So in this stage, where are we? So we are in ventricular systole now. Ventricles are contracting. So our first phase of contractions of the ventricles pushes these AV valves close, but there's not enough pressure yet to open the semiluminar valves. Okay, so they're still closed. But as we continue through the cardiac cycle and we get more of the conduction, now has gotten down to the apex, and we are entering, we're moving through more of the contraction. Now we have, we enter what's called ventricular ejection, because the pressure is now enough in the ventricles to blow open the semiluminar valves so the blood may move through. And now we're going to enter back to atrial diastole and into a period of isovolemic ventricular relaxation, same volume relaxation. So as our ventricles are relaxing, the pressure drops. And that's because blood is moving through those semiluminar valves and out. So the pressure inside is less in the ventricles because they're not full anymore. They've blown everything out already. So that gets us back to the beginning. So we have an overlap in events between the atria and the ventricles, which again makes it a little bit difficult to differentiate very specific events on the EKG. But nonetheless, it's an incredibly powerful diagnostic tool, and a lot of great information can come out of it. So at rest, a cardiac cycle is about 0.8 seconds, 0.8 seconds. So think about our average heart rate, right? 0.8 seconds, so 60 seconds in a minute, but it takes about 0.8 seconds. So you would expect our heart rate to be over 60. Now, the right side here is just showing the change in ventricular volume and pressure. And we start here. We're starting at point A, and we're moving this way now, opposite to what we looked at in the previous figure. So we have a relationship between pressure and stroke volume, which is the amount of blood associated with the cardiac cycle, with a single cardiac cycle. So we've got a couple terms we haven't talked about. We have an end diastolic volume and an end systolic volume. And the end diastolic volume is equal to the blood in the ventricles during this isovolemic ventricular contraction. And then we've got our end systolic volume is at this part of the figure in the cardiac cycle. Okay, so one other thing I'd like to point out off of this graph is we have two heart sounds, S1, which is heard here, and S2, which is heard here. So when the doctor is listening with a stethoscope, we can look at the EKG and figure out if those heart sounds are occurring at the right time. And say there's an extension, a longer period of time between the two heart sounds, between sound 1 and sound 2. Well, that tells us something's going on with the ventricular systole, since that's what's happening in the cardiac cycle at that time. All right, so the Frank-Starling law is about stroke volume. And what it states is that the stroke volume of the heart is going to increase in response to the volume of blood in the ventricles prior to contraction. That makes sense, right? It's the ventricles will fill up. We have enough pressure to open the valves so that the blood can move through. That's what stroke volume is. Okay, so there's a relationship between stroke volume and pressure. So these are some of the terms I'd like you to focus on throughout these two chapters, and I'll bring them up as we continue to go through. Okay, so our heart sounds, the sound 1 is called the lub, and it's associated with the closing of the AV, atria to ventricles, where S2 is the semiluminar valves. So different valves. S1 is the closing of the AV valves. That happens first. S2 is the closing of the semiluminar valves. That happens second. So looking at the kind of color coding here, we can figure out based on these sounds what's happening at different stages throughout the cardiac cycle. So we see this in effect more in lab because we can actually hear it and see it on the EKG that we do. So if we hear sounds of the mitral valve, you would expect, you'd put the stethoscope here. So it's showing you kind of where to place to get these different positions so that we can properly hear them. And this does take quite a bit of training to be able to hear them. And it takes a good stethoscope as well. You notice oftentimes cardiologists use a different type of stethoscope so that they can better hear these different sounds. So a murmur we would hear as a different sound. We hear, it sounds like a swoosh or a whooshing sound. Something like that. So what that could mean is that there's stenosis, and stenosis is narrowing. We can have stenosis in different parts of our body, but it always means narrowing. So stenosis of the neck is narrowing of the neck, which could press on the spinal column. So stenosis is going to affect resistance. It's going to increase resistance. And remember at the beginning of the lecture, we talked about the relationship between flow is proportional to the change in pressure divided by resistance. So if we hear the swooshing sound right before S1 or S2, it tells us which valve is affected. If we hear it before S1, well, that's the AV valve. If it's S2, those are the semiluminar valves. So just a good listen. A trained ear can get some insight about what is happening where in the heart, and specifically in what part of the cardiac cycle. Now, another pathology is regurgitation, which is also called insufficiency. And these can be related to a murmur. But what regurgitation is backflow. So the blood is not going in the right direction. So instead of blood moving from the atria to the ventricle, some of it's going backwards. Similarly, we can relate it to the S1 and S2 sounds. So if we hear that after S1, it's regurgitation of the AV. If we hear it before S1, it's stenosis of the AV. If we hear the sound after S2, it's regurgitation associated with the semiluminar valves. So a lot of good information can be determined just by taking a good listen. Now, I do want to emphasize that many heart murmurs are completely normal. There's a category of heart murmurs called flow murmurs that are not pathological. In fact, I have a flow murmur, but it doesn't mean anything. It just means that an experienced ear can hear it, but it doesn't present with any pathology. So flow heart murmurs are common, but aren't associated with pathology. All right, that brings us to what's called Wiggers diagram, which has a lot of information on it. I would never expect you to memorize Wiggers diagram, but I would expect you to extract information out of it if you're given it on an exam and ask questions about it. So let's put it together. We've actually looked at most all of this already in different segments already. Like we've seen all this before. We looked at this part of it when we were studying the conduction system. The top part of Wiggers is simply the electrocardiogram. Okay. The middle part is the pressure. I'm going to come back to that. The bottom part is the volume of the left ventricle. And then we've got the contraction and relaxation of the atria and ventricle outlined for you. So everything that is in this kind of darker peach color here is the atrial systole. Then we've got ventricular systole, ventricular disystole, and atrial systole, and you can see it tying in with the EKG. So atrial systole is P to the R. Now look at the pressure. Look at the volume. So the volume is highest prior to contraction of the ventricle. I hope that makes sense at this point. After the ventricle contracts, that's what pushes the blood out of it. So therefore the volume would be very quickly dropping off as we progress through the time associated with ventricular systole. And then the volume slowly increases and gets back to its highest point after atrial systole. So that's the relationship between the volume of blood. Now let's look at the pressure. And this is where it can be a little bit tricky to understand. So let's see, in this red line, we're looking at the left ventricular pressure, which is highest. Look at this, it's highest, leading to the systole. We need the high pressure to contract the ventricle. So it does make sense. There is a relationship between the pressure in the left ventricle and the blood, the volume of blood in the ventricle. The blue line is the left atrial pressure, and it's pretty constant. It's not changing too much. We've got in the black line is the aortic pressure. So what's changing most is the left ventricular pressure as we go through ventricular systole, ventricular contraction. So we've got our dicrotic notch shown right here at point B. Let me get back, excuse me, backing you up, sorry. At point B right here. I wanted to point that out. Where were we? Okay. Okay, so a little blow up of the bottom part of the Wiggers diagram shows the relationship between the EKG and the mechanical events of the heart. So our P is the atria depolarizing and so forth. We've gone through this already. You can, some of the coloring books that you can get on for physiology are really helpful for learning this. In fact, you can download coloring resources off of the internet for free. That might be a good way for you to understand and map up the mechanical as well as the electrical events of the heart. Putting in the heart sounds, here's S1 associated with the AV valves, S2 associated with the semiluminar valves. So our S1 sound is as the blood moves from the atria to the ventricle, where our S2 sound is as the ventricles contract and push the blood out through the semiluminar valves. Lub and dub, or dupe, I should say. Okay, tying in our volume, it is highest right before ventricular systole and is lowest after ventricular systole. And then it builds back up. All right, so looking at the events associated with our sympathetic stimulation versus parasympathetic. So the electrical conduction for the heart is an auto event. It happens right there in the heart, but certainly our nervous system can have an influence on it. So if we have the release of norepinephrine, it is going to lead to sympathetic stimulation of the heart. This is under control of a region called the Cardiovascular Control Center, which is in the brain, specifically in the medulla of the brain. So that's the integrating center. We are in the brain. Skeletal muscle is integrated in the spinal column. The control of heart rate is in the Cardiovascular Control Center of the brain. So we have sympathetic stimulation, norepinephrine is released. It binds to a type of receptor called the beta-1 receptor. These are present right on those autorhythmic cells. I mean, how else are we going to get them to fire quickly if we can't target those cells? So by binding of norepinephrine to the beta-1 receptor, this leads to the calcium, the sodium rush and the calcium-induced calcium signal, increasing the rate of depolarization, which increases heart rate. So it's going faster. Oppositely, look at this parasympathetic stimulation of the cardiovascular control center is with acetylcholine. Oh, we saw that before. But in skeletal muscle, it is a excitatory neurotransmitter. Different here. This is associated with parasympathetic stimulation. So it targets a different type of receptor, also present on the autorhythmic cell. And what it does is lead to a rapid increase in movement of potassium and calcium, causing hyperpolarization. So now we've dropped below our membrane potential and have a bigger distance to make up to hit threshold, which is how the heart rate is slowed under parasympathetic stimulation. So looking at cardiac output, we can determine a few different ways, but one way is the product of heart rate and stroke volume. So we just reviewed heart rate, and we can increase it through sympathetic stimulation with norepinephrine. We can decrease it with parasympathetic stimulation with acetylcholine. So we can affect that. But look how this ties in over here. So when we have sympathetic stimulation of the heart, we're with norepinephrine, we're also seeing an action with epinephrine too. I'll get to that in a moment. This causes the veins to constrict, which gets the blood back to the heart quicker, which is tied to stroke volume. Now, we have two pumps that can affect stroke volume, the respiratory pump and the skeletal muscle pump. What these simply are places in our body where our skeletal muscles push our blood vessels so that it flattens them down, that decreases the diameter, that increases resistance, and helps our blood get back to the heart. Because there's essentially no pressure or not much pressure in the venous system at all. So the sympathetic stimulation also affects the contractibility or contractility of the heart, which has an effect on the end diastolic volume. So cardiac output is the product of heart rate times stroke volume. All right, that wraps us up for chapter 14. I had to think about that for a minute, and I'll be back soon with your chapter 15 lecture. I hope you're having a great day or night, wherever or whenever you're seeing this. Take care. Bye-bye.